Right. So now we discussed the initial part of unit 2, right? So we discussed up to monosaccharides. Today we have to do disaccharides, right? So initial part of we did unit 1 in the first week. So second week we did first part of the unit 2, right? So before starting, I'll do a quick recap of what we have done till now, right? So go take the mind map of unit 2. Today I'll discuss from unit 2. So the basis of life. So in this lesson we discussed about chemical and cellular basis of life. So there are some few new students also. So this is the mind map, right? This mind map for each and every lesson, I am giving a note, theory note, short note, and a mind map. Theory note is the all the points which included in your resource book, which is bit different from the resource book because I categorize them separately. Because resource book it contains whole paragraphs, so it's difficult to understand by just reading the resource book. So I categorize then make a note. From note I make a short note uh, which is made of flowcharts. So the mind map is the third one. Mind map is a note with spaces. The same short note, I make it with spaces to make you easy to memorize the things, the points. So you have to, after doing the class, you have to go through this mind map each and every day, right? Each and every day means each and every day. You have to do it daily basis. Without doing it daily basis, it won't be easy to get a pass because there's the content of advanced level biology syllabus is way more than you can imagine, right? So some part when you are doing actually the unit 5 is a total year. It take total year to cover, right? So it's way more points are there. So to memorize it, you have to practice it from the beginning, right? So let's just go through the chemical and cellular basis of life, right? Let's discuss what we have done already. Chemical and cellular basis of life. The elemental composition, of living matter. Elements essential for life, there are 20 to 25% elements from naturally occurring 92 elements which are essential for life. Humans need 25 elements and plants need 17 elements, right? In living matter, 96% of body mass is oxygen, carbon, hydrogen, and nitrogen, right? Other 4% of body mass is calcium, phosphorus, potassium, sulfur, and other elements, right? Then we discussed about physical and chemical properties of water important to life. Here, water is inorganic molecule. It is major chemical constituent of living cells. It provides biological medium for living organisms. Water molecule is small, polar, and angular molecule. Polar means uneven charge distribution within the molecule. Oxygen is slightly negative and hydrogen is slightly positive. You should be able to draw the structure of water molecule. The space with the box with the space is given to draw the structure. So you don't have to draw it, just imagine it, right? You don't have to fill the blanks every day. Just Memorize the blanks. What are in the blanks, right? Then properties of water based on its chemical structure. Physical and chemical properties of water provide the ability to uh, render the vitality. Right. Then H bonds. Second page. H bonds are what are hydrogen bonds? Weak interactions between slightly polar or oxygen atom of one molecule and slightly polar hydrogen atom of adjacent molecule. Due to hydrogen bonds, what happened? Due to hydrogen bonds, properties of water occur. When water is in liquid form, hydrogen bonds are fragile. They form, break, and reform with great frequency. Properties of water to maintain the life on Earth. First one, cohesive behavior. Cohesion means attraction between water molecules. Addition means attraction between water molecules and other molecules. Due to cohesive behavior, there are two properties. What are they? Act as a transport medium. Due to cohesion and adhesion, water and it dissolves mineral transport as continuous column through xylem against gravity. And the high surface tension, next one is due to cohesion form water film, then water stakers can walk. Then second one, third page, ability to moderate temperature. Due to high specific gravity, organisms can absorb or release high amount of energy by slight change in their temperature. Functions as thermal buffer. It in temperature fluctuation, temperature is stable in aquatic bodies and living organisms. Then next one is high energy of vaporization. With minimum loss of water, can release much heat energy. Body surfaces maintain cool. Example, preventing from overheating the body. Human skin, sweating helps to maintain the body cool, and in plants, transpiration helps to maintain the plant cool and prevent overheating by the sunlight. Third one is expansion upon freezing. Generally, when temperature increases, density reduces. But when density reduces, temperature increases. In water, when less than 4 Celsius, it begins to freeze. Maximum density at 4 Celsius. Ice floats on water and aquatic organisms safe in winter, right? Then versatility as a solvent. Due to polarity, form hydrogen bonds with solutes. Solubility depend on polarity, not on ionic nature. Water soluble things are polar molecules like glucose, ionic molecules like sodium chloride, polar and ionic molecules like lysozymes. Right. Then we discuss about carbohydrates also, chemical nature and function of main organic compound of organisms. First one, carbohydrate. Carbohydrate characteristics. Most abundant organic compound. Elemental composition is oxygen, hydrogen, and carbon. Hydrogen to oxygen ratio is 2:1. Their general formula is CxH2OY. There are three groups: monosaccharides, disaccharides, and polysaccharides. Monosaccharides and disaccharides are sugars. Then we discussed about monosaccharides. Monosaccharides are the simplest form of carbohydrate. General formula is CH2ON. All are reducing sugars. All are sugars. Water soluble, crystalline form. Carbon molecules varied from 3 to 7. When there are 3 carbon molecules, we call it as triose. Example is glyceraldehyde. Phosphoglyceraldehyde is a derivative of glyceraldehyde. When there are 4 carbon molecules, we call it as tetrose. Example is erythrose, which is rare in nature. When there are 5 carbon atoms, we call it as pentose. Example, ribose, deoxyribose, and ribulose. RUBP is a derivative of ribulose. when there are 6 carbons, we call it as hexose. Hexose are glucose, fructose, and galactose. According to the carbonyl group, there are 2 types, aldoses and ketoses. Aldoses are glucose and galactose. Ketose is fructose. In aqueous media, some are in ring form. Right. That's what we have done in the previous days. Okay, so you have to go through the recording. Then you will understand what this is. Right. We did it fast, ne? So I explained it slowly, taking much time last week. Okay, so you have to go through the recording and catch it up. Okay, that's not very difficult. You will be able to understand by watching the videos. Okay, so now today we have to start from disaccharides, right? So what I am telling every day is you have to go through these mind maps. So if you go through the mind maps all the week in each and every day, you will find it very easy to memorize biology, right? If you don't go through it, you won't. It will be difficult. That's because the problem is in exam you will be asked from the resource book. They can ask from anywhere in the resource books. They don't ask things more which is not included in resource book. So you don't have to by heart things which are not included in the resource book. Earlier, when we are doing biology, there was no resource book. There was no resource book. So we have to find the syllabus. Actually, there was only the teacher's guide. So teacher's guide was the guide for us. So there was no limit actually. Several teachers teach several things. So then we didn't know whether this is important, whether this is not, or whether it is asking in the exam or whether it won't ask in the exam. We didn't know till the exam. That's how was it before 2016. Then 2016 they introduced this resource book, which started 2016, and the first exam group was 2018 group who sat for the exam, who had the resource book, right? So from then they don't ask questions outside this resource book. So you don't have to waste your time on studying things that not included in resource book. So you have to strictly adhere to this resource book, right? So then some students ask me that for some reason, I don't know why, there are more information about these monosaccharides, carbohydrates, and other stuff in O-level syllabus than in A-level syllabus. So when you go through your grade 10 O-level syllabus, O-level textbook, it includes more things than this in the first lesson. First lesson is molecular lesson. What is the main molecular constituent of life or something? It also contains these things. Monosaccharide, disaccharide, polysaccharide, we discussed in O levels. Here it is less elaborated in A level. Less points are there, but you don't have to then, as there are many points in O level syllabus, you don't have to by heart them O level syllabus, because you won't be asked from O level, so you will be asked from A level. I'm explaining this because one student asked me this. Okay, it's a genuine question. There are many things in O level. Why there are few things in A level? But actually, there are few things in A level only in this part. Right? Other parts contain way more than A level, O levels. Okay, so you just have to strictly follow the resource book. The problem in resource book is you can't understand it alone by reading it. That's the problem. That's because they wrote it like they make large paragraphs and you can't comprehend with these paragraphs. That's the problem. That's why I separately writing another theory note. Right? So in the class, we discuss the theory note, then the short note. At home, you have to go through the mind map each and every day. That's the plan. Okay, right. Now, let's start disaccharides, right? So we discussed about monosaccharides and carbohydrates. I'll quickly go through the carbohydrate and monosaccharide part. In carbohydrate is the most abundant organic compound in earth. In among organic, what is the meaning of organic compound? It contains carbon, right? When a compound contains carbon, we call it as organic compound. But actually, containing carbon is only a one feature. To become an organic molecule, it has to fulfill some features which we don't learn in biology. Just remember that if there is carbon, we call it as organic. If there is no carbon, we call it inorganic. But there are some compounds which contain carbon but inorganic, like CO2. CO2 contain carbon, but this is inorganic, right? It, the reason is there are several characteristics of organic molecules. To become organic molecule, they have to have all those characters, but carbon dioxide doesn't have those characters. Carbon dioxide have the character that it has carbon. That's the only character of organic molecules carbon dioxide has. So carbon dioxide is not an organic molecule. Among organic molecules, most abundant organic molecule is carbohydrate. That is the point you have to remember. Most abundant organic molecule on earth is carbohydrate. Then chemical composition of carbohydrates are oxygen, carbon and nitrogen. I'm sorry, and hydrogen. OCH are the chemical elemental composition of carbohydrate. That means all the carbohydrates are made up of oxygen, carbon and hydrogen, right? Then in right, in carbohydrates, always hydrogen to hydrogen to oxygen ratio is what? 2 to 1, right? Always 2 to 1. What is the general formula of carbohydrate? It's CXH2O. This have you learned this in O level also, ne? Right? So there are 3 types of carbohydrates. What are they? Monosaccharides, then disaccharides, and then polysaccharides, right? Right? Polysaccharides. These are the 3 types of carbohydrates. Among them, monosaccharides and disaccharides we consider that sugars. We consider monosaccharide and disaccharide as sugars, right? But we don't consider polysaccharide as a sugar, right? Last week we discussed about monosaccharides, right? So monosaccharides, පුතා මම වේගෙන් කරගෙන යන්නේ මම මේ කරපු ඒවා හින්දා හොඳේ. දැන් තේරෙන්නේ නැහැ වගේ හිතෙයි ගණන් ගන්න එපා. ඉතින් කරලා තියෙන එකයි. ආයි ස්ලෝලි කරන්න බෑ. ඔයා පොඩ්ඩක් ගිහිල්ලා අරක බැලුවම තේරෙයි ඔක්කොම. right? Right. Then monosaccharide is the simplest form of carbohydrate. Monosaccharide are the simplest form of carbohydrates, right? General formula of monosaccharide is CH2ON, right? Then all are reducing sugars. All are reducing sugars. Then all are water soluble. Reducing sugars, water soluble, and in crystalline form. These things you will learn in O level, crystalline forms, monosaccharides. Then there are about 3 to 7 carbon atoms in monosaccharides, right? When there are 3 carbon atoms, we called it as triose, right? When there are 4 carbon atoms, we call it as tetros. When there are 5 carbon atoms, we call it as pentos. When there are 6 carbon atoms, we call it as hexos. So you have to remember the examples for these things. There are a few examples given in the resource book, right? Then according to carbonyl group, there are 2 types. According to carbonyl group, there are 2 types of monosaccharides: aldoses and ketoses. Aldoses means it contains CO here. This format is aldose. Ketos, there's. So actually, you don't have to remember the format. No point. Okay. Right. Then aldoses. Aldoses are glucose and galactose. Ketos is fructose. Right? Then in aqueous media, most of the time in aqueous media, these monosaccharides are in ring form. That's why we draw glucose like this. In aqueous media, it is in ring form. Right. Now today, let's start to discuss about disaccharides. Right. In the first day, I told that the mono means 1. Di means 2. Poly means many, right? That's why we call carbon dioxide. What is the meaning of carbon dioxide? Dioxide ne kiyanne carbon dioxide. Why we call it as carbon dioxide? Because it has 2 oxygens. Carbon dioxide. CO2 kiyanne api carbon dioxide. Di means 2, right? Carbon monoxide, CO, carbon monoxide. Mono mean 1. Poly means many. Poly means many. Right? So now we are talking about disaccharides. Saccharides means sugars. Disaccharides, 2 sugars join together to form disaccharides. Right? All the disaccharides are sugars. Right? Then we discuss about disaccharides. Right? Disaccharides. Right? Disaccharides. Disaccharides means 2 monosaccharide molecules join together to form disaccharide. It's like this. There's 1 monosaccharide and here's another monosaccharide. These 2 form a bond and form disaccharide. All the disaccharides are sugars. We can consider disaccharides as sugars. So all the disaccharides are sugars. They are sugars, right? Then the bond which is formed in between 2 monosaccharide, we called it glycosidic bond. What is it? Glycosidic bond, right? Glycosidic bond, right? Glycosidic bond, right? So you should be able to write down the chemical reaction of formation of disaccharides, right? It's easy, Laman. Then most common monosaccharide what is the most common monosaccharide you know? It's hexoses. Hexoses blue. In O level we discussed about only in O level, what are the monosaccharide you learned about? Glucose, fructose, and galactose. In O level, they are talking about those three only. So they are the most common ones. But in here, we discuss about trioses, tetroses, pentoses, and also about hexoses, right? Here, you should be able to write the chemical reaction, right, of formation of disaccharides. Disaccharide formed by what? From two monosaccharides. So as monosaccharide, we take hexose here, right? Here, two hexoses means C6H12 and O6 plus another C6. Right. C6H12 and O6 plus another C6H12 plus O6. What happened? Condensation reaction occurs and water molecule will go out from this, right? I'll show you what happened, right? Anyway, then when the condensation reaction occurs and forming a glycosidic bond between these two molecules, what happens is water molecule will go away from these two, right? So there should be a water molecule coming out from these two monosaccharide molecules. So what happened is, now usually what should happen? There are two organic compound, two hexoses. So when they combine, what should happen? Carbon should double. Hydrogen also should double. Oxygen also should double, right? Then when carbon 6 plus carbon 6 means there should be 12 carbons, right? So there should be 12 carbons, right? 12 hydrogen and 12 hydrogen means there should be 24 hydrogens. But what happens is there are two hydrogens which went to make water molecule. So from 24 hydrogens, now there are only 22 hydrogens. Two went to form the water molecule. And oxygen, there should be 12 oxygens, but one went to form the water molecule. So now there are 11 oxygen molecules. So it's easy, Laman, right? You just have to double the monosaccharide and remove a water molecule from it. Then you will find you can have the formula of disaccharide, right? Actually, this is not valid for all the disaccharides, Lam. This is valid for disaccharide made up of hexoses, like glucose, or fructose, or maltose, maybe glucose, fructose, galactose, like that hexoses, right? But anyway, you have to remember this equation. You have to remember this equation, right? Right. So then I'll show you how the glycosidic bonds will form, right? So in aqueous media, how a monosaccharide is there? Monosaccharide is in which form? In aqueous medium, monosaccharides are in ring form. Ring form means in when we are considering glucose, we know glucose, right? When glucose is in dry form, that means in the packets, glucose when the glucose are in dry form, they are linear molecules. Linear means they are like this. The carbon, 6 carbons, 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 6 and hydrogens, oxygens, everything is there. They are linear molecules, right? When it is in dry form. But when they are in aqueous media, they form a ring structure like this, right? So in aqueous media, monosaccharides are in ring form, right? Yeah. I'll show you how a glycosidic bond will form. What happens is when there is two monosaccharides, right? Here's one monosaccharide. It's in ring form. Here's another monosaccharide, which also in ring form in a aqueous media. In these monosaccharides, there are hydroxyl groups, OH groups. Here's a OH. Here also there's a OH, right? OH means what is here? There's a carbon which bind with carbon and carbon like this. It's polymers. Then here's a O and here's a H, OH. And here's OH. Like this, there's a point where there is a OH group, right? So this OH group, there are two OH groups in these monosaccharides, right? From this OH group, a water molecule will leave these two molecules like this. One H from one monosaccharide and a OH from the other monosaccharide will leave, forming, doing a condensation reaction. We call these reactions condensation reactions. What happens when the water molecule is removed, right? One H from here, one OH from the other monosaccharide, there will be a formation of what? A bond which is called as glycosidic bond, right? So the oxygen be coming to the middle and forming a glycosidic bond. This is the glycosidic bond. This is how a disaccharide is made, right? OH from one molecule and OH from other molecule. OH from one molecule and H from other molecule will leave to form a water molecule, right? Then there are three examples for disaccharides which we discuss in the lesson. They are same as the O-level syllabus: maltose, sucrose and lactose. Right? Maltose is made up of two glucoses. Glucose plus glucose is what is maltose plus water, right? Glucose plus glucose is maltose. It's easy to remember, glucose, maltose. हम කියන්නේ හැමතිස්සෙම එකම જાතිය දෙන්නෙක් එකට එකතු වුණොත් maltose තමයි කොහොමද? හරි? Glucose plus glucose, maltose, right? Maltose නේ. හරි? Then glucose plus fructose. Glucose plus fructose. These are very simple things, okay, in biology. That's why I just telling there are many complex things in the near future. So just you just have to memorize these points only. Don't by heart other things, right? There are many more things that you can learn about these monosaccharides and disaccharides, but don't try to go beyond the resource book, right? Try to be within the resource book, because in the exam you will be asked question from the resource book, right? So you just follow the resource book, okay? And you have to by heart every single point in the resource book. That's the problem. They can ask from anywhere from the resource book. And they, in the essay questions, what is the problem? They might just ask, write a short note about disaccharides, right? They just ask the question as, write a short note about disaccharide. What should you write? You should write everything in the, which are in the resource book, right? This is all in the resource book. This is the points which are in the resource book. So you have to write all these points, right? For each point, they'll give marks. If you miss a point, you will not get the mark. That's how biology works, right? Not like other subjects. Then physics, if you write the answer, you will get all marks. It's easy to take marks at physics, because if you know to do the maths and solve the question and mathematical parts and the question, then they have to give you all the marks. But in here, what happened is, they ask broad questions and they give points. So you have to by heart and memorize and write down every single point, right? Sometimes they decide this point is not, no need to give marks for this point. They'll remove these points from the marking scheme. So can't help Puta, right? You just have to write everything. It's the safest way, right? So you have to write the points which are in the resource book. Those points are the points which I used to make this short note and mind map. So you just have to go through these mind maps. If you can memorize all the points in mind map, then you can write all the point in the exam, right? So they can't reduce your marks. So the thing is, you have to memorize all the things in resource book. That's the thing, right? It's a bit difficult. Difficult because the load, not of the theoretical parts are not that much difficult. The problem is load is high. The amount you have to memorize is way more than you can imagine. So you have to, what I tell is, don't let your brain forget what you learn. That's the problem. What's happening is, I have told this every single day, but I'm telling it again because this is the most important thing. What happens to you students are, you start very well, right? In every school, in every class, in the beginning, you start very well because it's the beginning. So every time, we start well, but most of the students don't continue it, right? So you have to be disciplined. You have to continue the things, right? නිකම්ම ළමයි මේක නිකං එකපාරක් පාඩම් කරලා ගොඩ දාගන්න බෑ O level වගේ A level. You have to understand this is two separate entities. You can't do A levels like O levels. And you have O levels can be done very easily. O level science means nothing. There are more subjects, more lessons, 35 lessons are there. In biology, there are only 10 units. That's true. But those 10 units contain way more content than O level syllabus, right? So the problem is, when I'm teaching you these contents, you can understand it. That's okay. It's easy. Most of the parts are like this. They can be, they are understandable, right? Understandable, but not memorizable. That's the problem. You will forget it. The exam is in 2 years. So what you learn here will ask in the exam from the unit 1, from unit 1. So you have to keep those points in the memory till 2027. That's the problem, right? That's the problem in biology. You have to memorize them. You have to by heart them. To by heart them, I made an easy way to by heart them. That's the mind map. So you have to just go through the mind map each and every day, right? Then don't fill the blanks, right? Just go through it. Just fill the blanks from your mind, right? Don't write anything. Just go through it. If you can't fill that blank, just go through the short note. If you can't understand the short note, then go through the note. Then you will understand, right? So you just have to what? Go through the mind map every day. Start from today. Start it from today. If you are new to the class, so you have to start from today. Go through the mind map. Go through the mind map. Each and every day, go through the mind map. It will take only 10 minutes, right? Still, there are very few. Today, I go through the mind map before starting the class. It took only 10 minutes, 10 to 15 minutes. So, to you to go through the mind map, it will take only 10 minutes initially, right? When you are practicing, the time will reduce more. So, you have to give that 10 minutes for biology from the beginning, each and every day, right? පුළුවන් නේ? දරනවනේ? හරි. දවස් හැමදාම කරන්න ඕනේ පුතා. නැත්තම් මේක මිස් වෙනවා. මේ හැමදාම කියන්නේ ඒකයි. මේක මිස් වුණොත් ඒ එකතු වෙන ලෝඩ් එක දරා ගන්න බෑ. හරිනේ? ඔයා දැන් මේ සතිය කරන ටික ලබන සතිය වෙද්දි හැමදාම බලලා අර හිස් තැන් ටික හිතෙන් පුරවලා ආවේ නැත්තම් තව සති දෙකක් ඊළඟ සතියේදී ඔයාට මතක නැහැ කිසි දෙයක්. එහෙම තමයි මේක. මේ හැටි ඉතින් අවුරුදු දෙකක් යද්දි කොයි මතක තියෙන්නේ? පෙරේදා කාපු එකවත් මතක නැහැ හරියට නේ. එහෙනම් අවුරුදු දෙකක් යද්දි මතක තියෙන්නේ නැහැ ළමයි. ඒකට තමයි අපි මේ ප්‍රැක්ටිස් කරන්න ඕනේ මතක තියාගන්න. easiest way is not to forget. අමතක වෙන්න දෙන්න එපා. If you forget, you have to work hard again to memorize it. So don't let your brain to forget these points. Just by heart this point by reading the mind map each and every day, right? Glucose and fructose combines to form what? Sucrose plus water molecule. Then what are the other two? Glucose and what? Galactose. Galactose form what? Lactose. Lactose plus water. This reaction, we called it as condensation reaction, right? In condensation reactions, water molecule is formed from two molecules and forming a bond. This bond is called as glycosidic bonds. So the bonds in carbohydrates, bonds between monosaccharides are glycosidic bonds. You have to remember the name, glycosidic bonds, right? Right. There. From these three, two are reducing sugars. One is non-reducing sugar. So there are some practicals also which related to the syllabus. So we will discuss it later. Okay. So I think you can remember the practical which done to identify disaccharides in O level. Can you remember what we used to identify monosaccharides? Benedict solution. Can you remember how it done? You take a sugar, you take a water, glucose solution, then you put what? Benedict solution. Benedict. It's blue color. Benedict solution. Then you boil it in water, and what happened? The color will change. Blue will change to blue become green. Green become yellow. Yellow then orange. Then bright red color deposition will occur. We will discuss it later. I just remember that in O levels you did this. We called it Benedict test. We used it for what? To identify monosaccharides and some disaccharides, right? So what happened in this chemical reaction is these sugars are reducing agents. So they will reduce the Benedict solution. Benedict solution is blue colored. When it is reduced, it formed bright red color deposition. Can you remember that sucrose doesn't do this reaction? Sucrose, when Benedict solution is added to sucrose and boiled, this color change won't happen in sucrose. Then what we did, we put acid, hydrochloric acid or sulfuric acid or any acid, small amount of acid, and then then heat it. What happened there was, then it shows bright red color deposition. What is happening there? What's happening there is, when when a sucrose, here there is sucrose. Sucrose is like this. It's made up of glucose and a fructose, right? Glucose and fructose, right? This is non-reducing sugar. Sucrose is non-reducing sugar. So it won't reduce the Benedict solution. That's why it won't give a bright red color deposition, right? But all the monosaccharides, glucose, fructose, galactose, and other two disaccharides, maltose and lactose, all 5 monosaccharides and disaccharides are reducing agents, reducing sugars. They will reduce the Benedict and form a bright red color deposition. But sucrose is not a reducing sugar. That's why it doesn't give bright red color deposition. Right? This is the practicals which you done in O levels. Right? But what happens when you put acid to it, sucrose? When you put acid and boil it, what happened is, they will break down this glycosidic bond. Right? Then what is in this solution now? Is there any sucrose? No. Sucrose will break down into glucose and fructose. Now what happens? Glucose and fructose are reducing agents, reducing sugars. It will reduce Benedict solution and give the red color, bright red color deposition. Right? So, what is the take home message from it? You just have to remember that sucrose is non-reducing. Sucrose is non-reducing sugar, and maltose and lactose are reducing sugars. Right? Sucrose is a non-reducing sugar. Right? Now go to take the short note, Puta. Take the short note. In short note, right, read the disaccharide part. Right? I'll wait a minute. Right? Right, unit 2, part 1, short note. Here, go through the short note. Disaccharides. In disaccharides, all are sugars. All the disaccharides are sugars formed by joining of two monosaccharides by glycosidic bond. Two hexoses, 2C6H12O6 goes through a condensation reaction to form C12H22O11. It's disaccharide. Then here's how the glycosidic bond forms. It removes OH from one monosaccharide and a H from other monosaccharide. Then types of disaccharides. Glucose plus glucose, it is maltose. Glucose plus fructose, it is sucrose. Glucose plus galactose, it is lactose. Reducing nature, reducing sugars are maltose and lactose. Non-reducing sugar is sucrose. That's all you have to keep in your memory about disaccharides. Actually, in O level, it contains more than that in disaccharides. Ne? You have to, I had some more things are there. Right. So just you have to remember, memorize all these points, Puta. That's the problem in biology. You have to memorize all these. For that, you will be helped by that mind map. So go through the mind map each and every day. Try to fill the blanks each and every day. Right? So I always told you that when you are practicing something new, then you have time ne Puta. You have time ne. Time තියෙනවා නේ මොකුත් කරන්න නැහැ නේ දැන් නේ. So you are doing this ne. So I think you all are doing biology to with the plan of becoming a doctor, right? Most of you want to become a doctor. That's why you doing biology. Most of you, not all, actually not all. Some have some plans, but most of the students who are sitting for advanced level in biology stream has the dream of becoming a doctor, right? That's the dream. So there are about 56,000, 57,000, around 60,000 students who are sitting for the exam, biology, in a year. In each year, around 60,000 students sit for biology. But only around 2,000 students will be selected to medical faculties, right? So it is a competitive exam. Actually, the most competitive exam is advanced level in Sri Lanka, right? It is the most competitive exam. Only 2,000 will enters the medical faculty from 56 to 57, 60,000 around, right? So it's a competition. So it's a competitive exam. So competition means you have to work harder than others to enter. So in this race, the first 2,000 will enter to the next stage. That's what happened. So you have to run faster than others. That means you don't have to push others to go there. Some people are doing that also. Sometimes even in our schools, even in our class, even in our days, some students don't let others study. That happens. That's the human nature, right? It's everywhere. So you don't have to do that. You just have to work hard, right? In our schools also, there are some when someone become the first place in the class, the second place one will actually I became the first place in all section in first term test in grade 12. The one who was first for several years in my O level class also stopped talking with me. What the hell, man? That's how it happened, right? So some people are try to push others down and go up. That's not the way, right? You just have to work harder than others to go enter to medical faculty, right? So if you are willing to go to the medical faculty, you have to work hard from the beginning, right? So I am teaching you the pathway that lead you to medical faculty easier than others, right? This is the easiest way. So easiest way is not letting forget, right? That's the way, easiest way. If not, what happen? You today we learn this and you will buy hard all these things and you will again you learning this in just before the term test. What happened? You won't remember single thing. Then what you have to do? You have to buy hard it again. You have to work hard again. So that's waste of time and waste of your energy. Just learn things and memorize things in the class, then don't let it forget, right? That's the way. So if you are planning to go to medical faculty, if you are want to become a doctor, you have to work hard from the beginning. You have to work not hard actually. You have to have the discipline to work every day, right? So as I told, every day when you are applying something new to your, if you are applying something new to your life, it's very difficult ළමයි. දැන් කියන්න ඕගොල්ලෝ උදෙන්ම නැගිටලා උදේ 4ට නැගිටලා හැමදාම දුවනවා කියලා හිතාගත්ත කියලා. එහෙම හිතනවනේ, හිතලා තියෙනවන්නේ. පුතා හිතුවක් දන්නේ නැහැ. අපි නම් එහෙම හිතලා තියෙනවා. උදෙන්ම නැගිටලා දුවනවා කියලා හිතනවා. හිතනවා විතරනේ ළමයි. ඔක් කරන්න හරි අමාරු. Right? Because it's new. හැබැයි if you do it for more than 21 days, that's a fact. When you do it for more than 21 days, it will be a habit. Then it's easy for you to continue it. This is also like that. It's very difficult to start, very difficult to begin. But if you start it and continue it, you can continue it throughout these 2 years, right? Just follow what I'm telling. That would be enough, right? That would be enough. You just have to go through these mind maps only. So I told you නේ හවසට තාම්මත් ඒක දෙන්න වෙලාවට mind map එක බලන්න. Right? You have to combine it with something you are already doing. Right? You are already drinking mother's tea at the evening, mom's tea. So each and every day when mom give you the tea, you just go through the mind map while drinking the tea. ඒක බොන වෙලාවේ තමයි යන්නේ. හරි උණුවෙන් තේක දුන්නොත් තේක නිවෙද්දි මයින්ඩ් මැප් එක බලලා ඉවරයි. නේ? ඉච්චර ගොඩක් නෑ මුලින්. හරිනේ පස්සේ එකතු වෙද්දි it won't be a problem. Why? Because you are practiced to go through the initial part very quickly. On the first day it will take 10 minutes. On the second day it will take 9 minutes. When you are practicing for a month, it will only take 2 minutes to go through the mind map. The same part, right? Like that, just go through the mind map each and every day. And each and every day means each and every day, not every other day, right? Still you have to go through each and every day. I'll tell you when to reduce the frequency. Then later you can reduce it slowly and once a week is enough. It will be later. Then we are making another note from the mind map. I'll teach you later, right? We can make a topic note from mind map. I'll tell you later. You don't have to buy hard all these things now. Then you will be, it's very easy, lamai. It's very easy. You just have to keep the things in your memory. Don't forget them. If you forget, that's the problem. Don't let your bloody brain to forget these things, right? The brain is always trying to forget things, right? The brain don't want these things. The brain functions in a different way. They keep in the memory what is important for you and what you like actually, right? Try to keep in your memory what you like. Do you like these things? Some people might be there who likes doing these things, but most of the people, you and me also, I don't also like these things and studying, but can't help me, right? So won't let forget, right? Keep in mind, go through the mind map each and every day. I can assess you who are in the physical class, but mainly in online students, I can't assess you, Nibuta. So you have to do it yourself. Keep the discipline, right? You have to do it yourself, right? Then you will see how effective this mind map is when you're doing the first term test at your school. You will see how your marks will be, right? With comparing to the other students, right? Just go through mind maps, these mind maps. Tell your friends also. Give them those mind maps. It's okay, right? Tell your friends, teach your friends what you have learned. It will keep in your mind way more than you learn alone. When you teach someone, it will be in your mind for longer duration than just reading yourself, right? Just try to teach someone, right? Okay. Now let's talk about polysaccharides. What are polysaccharides? Poly means many. So many sugars combine to form a polysaccharide. Many sugars. One sugar, monosaccharide. Two sugars, disaccharide. Many sugars, it's polysaccharide. Right. Okay. Polysaccharides. Saccharides means sugar, right? Polysaccharides. Polysaccharides. Right. In polysaccharides, you have to remember a few characteristics of polysaccharides. Few characteristics of polysaccharides. Polysaccharides are the macromolecules. What? Polysaccharides are macromolecules. What is the meaning of macromolecule? Macromolecule means it should be large. Macro means large, micro means small. Then they should be large molecules. It's compatible. Polysaccharides means many saccharides. Then it should be a macromolecule. To become a macromolecule, there should be two characteristics which you have to learn. One is to become a macromolecule, your molecular weight should be more than 10 to the power 4 to 10 to the power 5. If your molecular weight is more than 10 to the power 4 to 10 to the power 5, we consider it as a macromolecule. And to consider as a macromolecule, it should be a polymer. It should be a polymer. What is the meaning of polymer? What is the meaning of polymerization? මතකද ඕලෙවල් වලට කියන්නේ polymerization. Polymerization means you have to combine, you have the units, subunits have to be joined like this in a linear sequence, right? Like many subunits joined together linearly. This is called polymerization. So when it is formed a molecule which subunits are joined together and form a linear molecule like this, we call these as polymers. So polysaccharides, polysaccharide means many monosaccharides joined together. So this is a polymer. This is a biopolymer. Biopolymer means polymers which occurred in organisms. So this is a biopolymer, right? So to become a macromolecule, this molecule should have a molecular weight more than 10 to the power 4 to 5, and it should be a polymer. And right, this, then the second one is these macromolecules are, sorry, these polysaccharides are biopolymers. What's the meaning of biopolymers? I told you, polymers which made in organisms. They are biopolymers. So here, to form monosaccharides, there might be few hundred to few thousand monosaccharides. There should be few hundred to few thousand monosaccharides, right? To form a polysaccharide, right? 100 to 1000, like this. So 100 to 1000 monosaccharides join with which bond? Here, it's glycosidic bond, same bond, right? Form glycosidic bonds and form a polysaccharide, right? Few hundred to 1000 monosaccharide. We call these subparticles monomers, right? In here, monomer is glucose. Monosaccharide is the monomer. This is the polymer, right? These subunits are identified as, we call them as monomers, right? Here, the monomer is monosaccharides. Few hundred to 1000 monosaccharides joined together by glycosidic bonds forming polysaccharides, right? Then these polysaccharides, when remembering the characteristics of polysaccharides, you can keep in mind the polysaccharide what you know the most. What is the most common polysaccharide you will find in your day-to-day life? Starch. Starch is the most common polysaccharide which you find in day-to-day life. So, if you can remember starch, you can remember the characteristics of polysaccharides, right? Starch is, it is sweet. Is it sweet? Starch? No. Monosaccharides, glucose, it's sweet. Disaccharide example is sucrose, means sugar, which we use in day-to-day life. It's sweet. But starch, is it sweet? Then it is not a sugar. That's why it's not sweet. It's not a sugar. Not a sugar. In the beginning of the lesson, we told that in carbohydrates, there are 3 types: monosaccharide, disaccharide, polysaccharide. We considered monosaccharide, disaccharides are as sugars. We don't consider polysaccharide as a sugar. You can remember, in the beginning we discussed, right? So, polysaccharides are not sugars. They are not sugars. Does starch dissolve in water? No. So, polysaccharides are not water soluble. Polysaccharides are not water soluble, right? Then, does starch is in crystalline form? No, it's a powder. Sucrose is a crystalline form. Glucose is powder or crystalline. It's crystalline. It's small crystals. Just like powder, just take sugar to one hand and starch to one hand and just do like this. Then you will see what is a powder, what is a crystalline. Glucose is small crystals, very small crystals, but starch is a powder. So starch is, what is starch? It's not in crystalline form, not crystalline. not crystalline, not in crystalline form, right? These are the characteristics of polysaccharides. So polysaccharides are made from monosaccharides, right? Poly means many. What are the characteristics? They are macromolecules. Their molecular weight is more than 10 to the power 4 to 5, and they are biopolymers. They are polymers. These are biopolymers. Few hundred to thousand monosaccharides join together to form polysaccharides. They are not sugars. They are not water soluble. They are not in crystalline form. Right? Right. Then there are two types of polysaccharides, right? There are two types of polysaccharides. What are these two types of polysaccharides? We can categorize them based on their structure and based on their function, right? We can categorize polysaccharide based on the function and based on the structure. Types based on function and based on structure. Based on function, there are two functions that polysaccharide mainly do. One is storage, one is structural. So there are storage polysaccharides, as well as there are structural polysaccharides. So you have to remember the examples for storage polysaccharides and structural polysaccharides. What are the storage polysaccharides? What do you know? Starch. Starch is the storage polysaccharide of plants. Plants ne starch stored karan, right? So starch is the storage polysaccharide of plants. Starch. Then what is then glycogen. Glycogen is the storage polysaccharide of animals and fungi. Both store glycogen, right? So why storing them? Why are we storing these polysaccharides? Why? Because monosaccharides are our main energy source, right? Main energy source is monosaccharides. So what happened is, when you eat, let's see, you will eat half of a bread. Half of a bread. You eat. So what is in it? It's starch. What happened is, in the digestive system, it will be break down into, starch is a polysaccharide. It can't be absorbed directly because it's a large molecule. So what we are doing is, we break down it to what? To monosaccharides. What is the monosaccharide? Formed from starch, glucose. Then the glucose will absorb into the body, right? Into the bloodstream. Then what happened to the glucose absorbed into the bloodstream? So is it does our body needs all the glucose at once to make the energy? No, we ate half of a bread. It's a very huge amount of glucose is coming to the circulation, right? But what happened is, some of those glucose will use to form the energy. Breaking down of glucose done in cellular respiration. We'll learn it in this lesson, this unit. We discuss about cellular respiration also, right? So we break down the glucose and form energy, right? That's what happened in cellular respiration. So what happened is, From those glucose, some amount is used to produce energy. The other glucose, what happened? They'll be stored to use later. Where, as what it stored in us, it stored as glycogen. What happened is, from glucose they form a polymer called glycogen. Right? From monosaccharide they form a polysaccharide called glycogen and store it in our liver and muscles. Right? Then what happened when we don't eat all the day, nilamai. So we eat a pang, bhagyak, and then go on a try, go on a walk. What happened is, there's a dog who started to follow you. What happened? Barking and not following like, you know what I mean? Hey, they'll bite me. So what you do? You will run. What happens? You need energy. From where the energy comes? First, from the blood glucose which is in the blood circulation. They'll break down the glucose and form energy. But within few minutes, what happened is, the glucose within the blood circulation will reduce. Then what happened? Then you need glucose to produce energy. From where the glucose come? From these glycogens. It is stored in the muscles, in muscles and liver of our body. What will happen is, they'll break down the glycogen and release glucose to the bloodstream, slowly releasing glucose. It's a polysaccharide. When you break down that glycosidic bond, what happened? Monosaccharides are released into the bloodstream. Right? So, why we are storing glycogen? To use later as energy source. Right? Okay. So we can categorize polysaccharide according to the function as storage polysaccharide and structural polysaccharide. Storage polysaccharides, starch and glycogen. What are structural polysaccharides? Cellulose, you know cellulose. Cellulose in cell wall of plant cell. Then hemicellulose, also a polysaccharide. Cellulose, hemicellulose. Then also pectin. Where do you find pectin? Pectin is in middle lamella. What is middle lamella? Do you know middle lamella? Middle lamella is the structure. It's not actually structure. It's an area where two adjacent plant cells held together. Right? The plant cells have a cell wall. The plant cells have cell wall. Right? This is two plant cells. In between these two plant cells, there is a sticky material which stick these two plant cells together. This is called middle lamella. This is a sticky it is a sticky polysaccharide rich structure. Right? It's a sticky polysaccharide called pectin, calcium pectate and magnesium pectate. We'll learn about these things later. Okay? In separate lessons, we are learning about these things. So don't worry. Right? So middle lamella, it is made of pectin. Right? Pectin is a polysaccharide. Right? Pectin, calcium pectate and magnesium pectate. It's a sticky polysaccharide. It's like a glue. It glues two cells together. Right? Right. So here you have to remember the examples for storage polysaccharides and examples for structural polysaccharides. And it's easy, right? Then what are there are types of polysaccharides based on the structure, right? Based on their architecture. Based on their architecture or structure, architecture. According to the architecture, there are linear polysaccharides and branched polysaccharides. There are some linear polysaccharides and some branched linear polysaccharides and some branched polysaccharides. What are the linear polysaccharides? Cellulose and amylose are linear polysaccharide. Cellulose and amylose are linear polysaccharide. Hemicellulose is a branch polysaccharide and glycogen also a branch polysaccharide. Branch polysaccharides are hemicellulose, then hemicellulose is a branch polysaccharide, and then glycogen is a branch polysaccharide, then amylopectin is a branch polysaccharide, right? Here, what is the meaning of linear? Linear means they like this. Polysaccharides are like this, just linear, no branches. In glycogen, it's like this. In glycogen, it's like this, it's branched. It's branched, right? Now, the types of polysaccharide based on its function, there are two types: storage polysaccharide and structural polysaccharide. Based on the architecture, there are two types: linear and branched ones. Right? තේරුණාද? ප්‍රශ්න තියෙනවද? නෑ නේ මේ ටිකේ ප්‍රශ්න තියෙන විදියක් නෑනේ. අනේ ප්‍රශ්නයක් තියෙනවනේ මේ වෙලෙම අහන්න හොඳේ. So don't go home with questions in your mind. Just ask. and අහන්න පුළුවන්. එතකොට ප්‍රශ්න විසඳගෙන යන්න. Right. Then there are some examples for polysaccharide. Few examples for polysaccharides. There are few examples for polysaccharides. You have to remember. What are those examples? So you have to remember the polysaccharide and what it was made up of. That means the monosaccharide used to make those polysaccharides and its function. So first examples for polysaccharides. First one is starch. Starch is made up of glucose. What is the function of the starch? It is stored in plants. Stored in plants. First one is starch. Second one is glycogen. Glycogen also made up of glucose. It's stored in animals and fungi. තමයි මේ පාඩම් හරි බෝරින් පාඩම් ඉතින් කරන්න දෙයක් නැහැ. හොඳේ. There are way more interesting පාඩම්. හරි මේ lessons in here. And this is the first lesson. Boring ඉතින් පොඩ ගන්න polysaccharide, polysaccharide, චූඩි තේරෙන්නේ නැහැ, පේන්නෙත් නැහැ. ඉතින් ඔය ටික මතක තියාගන්න. The problem is most of these examples are new to you. So these words are very difficult for you to memorize. You'll see. But don't worry, because most of them we'll learn about most of them in the future. Right? These examples we meet in the future, like pectin. Middle lamella, pectin is in middle lamella. You don't know middle lamella. But in when we are discussing about plant cells, we discuss about the middle lamella. Right? Then you will know that middle lamella is this middle lamella is made by pectin. We discuss separate subtopic about middle lamella. Then you will rememorize. Like that, then when we discussed about monosaccharides, there are trioses, glyceraldehyde. The phosphoglyceraldehyde is a derivative of glyceraldehyde. So it's very difficult now. You will be like surprised, what the hell? This glyceraldehyde, phosphoglyceraldehyde, then many words, don't worry. You will see them in the future. In separate lessons, you discuss about these phosphoglyceraldehydes and other things in later. So then you will remember this. But just now, for now, just by heart those names. Okay, for just now we'll discuss about them later, right? So glycogen is made up of glucose stored in animal cells and fungi. Then cellulose. Cellulose also made up of glucose. It is in cell wall. Cell wall. Cell wall of what? Plants. Cell wall of plants, and also in chlorophytes. Chlorophytes, who are chlorophytes? Chlorophytes, they are not plants. They are protists. You know protists? Can you remember? We divide organisms into three domains, bacteria, archaea, and eukarya in O-level syllabus. In bacteria, archaea, eukarya, we divide into 4 kingdoms. What are they? Protists and fungi and plants and animals. These are the protists. There's a type of protist called as chlorophytes. They are protists. We'll discuss it later. Okay? Just remember, these protists also containing a cell wall of made up of cellulose. Right? Just remember. Right. You can remember these things, in all level. So just remember there is a protist like amoeba paramecium. There's another one called chlorophytes. Right? පාසි කියන්නේ ළමයා. පාසෙවල algae පොඩට වැටෙන්නේ මේවා. Type of algae, green algae is chlorophyte. There are many types of algae. We learn about them in 3rd unit, right? Then so don't worry, you have to learn them. Right? Right. Then cellulose is made up of glucose. It's in cell wall. Then another one is inulin. Can you remember inulin? It was in the knowledge. No, not in knowledge syllabus. Inulin. Actually, this is not that much important, Puta. The problem is the inulin is made up of what? Fructose. It is made up of fructose. Right? It is made up of fructose. So, what is the important? This is the only polysaccharide which is made up of fructose. That's why we are learning it. Right? It is the storage polysaccharide of Dahlia tubers. there's a plant called dahlia. In dahlia tubers, normally plants store polysaccharides as starch, right? Normally plants store carbohydrates as starch. But these dahlia tubers, they store carbohydrate as inulin, which is made up of fructose. That's why you have to remember it, right? In storage polysaccharide in dahlia tubers. Dahlia tubers, they store inulin as the polysaccharide. Then pectin. Where is the pectin? We discussed. Pectin in middle lamella, ne? Pectin. Pectin is made up of galacturonic acid. What is the monomer? Galacturonic acid. Galacturonic acid. This is the monomer. Just like glucose, it's the monomer. It's a monosaccharide. Galacturonic acid. It's a monosaccharide. Right? It is the one which is polymerized to form pectin. Right? It's in where is the pectin is? In middle lamella. It's in middle lamella. Right? You can remember the middle lamella. Ne? Middle lamella is the structure which stick the adjacent cell walls of plant cells. Right? Right. Then hemicellulose. Hemicellulose is in cell wall, ne? Hemicellulose. It is made up of pentoses and hexoses. Just remember as pentoses and hexoses, that enough. Okay. Pentose and hexose. Where it is? It is in cell wall. Cell wall of plants. Right. Then there's another one called as chitin. What is it? Chitin. Chitin. This is a special polysaccharide. Why it is special? It contains nitrogen. It contains nitrogen. So what are the chemical composition, elemental composition of carbohydrates? OCH. Only OCH. It doesn't contain nitrogen. It just contain oxygen, carbon and hydrogen. But in here, in this chitin, there are nitrogen atoms. So this is a nitrogen containing polysaccharide. This is a nitrogen containing polysaccharide, right? So it is made up of a monomer called galacturonic acid. What is it? Sorry. It's made up of a monomer called glucosamine. Glucosamine. So you don't have to buy hard. Amine means, what is amine? Ammonia. What is ammonia? Can you remember? NH3 ammonia. Amino group. What is the amino group? In amino acids, can you remember? Amino acids, there's carbon, R, this is all level, hydrogen, and there's a carboxyl group, COOH. There's a amino group, NH2, ammonia. Here, amine means there's a nitrogen. All the amines have nitrogen. So the monomer which made chitin is nitrogen containing monosaccharide, polysaccharide. It's the name of this monomer is what is glucosamine. Amine means nitrogen, right? Though the monomer is glucosamine. අමුතුවෙන් මතක තියාගන්න දෙයක් නැහැ නේ. Glucosamine. So most of the time you confuse galacturonic acid and glucosamine. That's what happened. Okay? Pectin is from galacturonic acid. Chitin is made up of glucosamine. Just like I shuffled it up in the beginning, you will also shuffle it up in the exam. So don't shuffle it up. Just remember that amine means nitrogen. So it is made up of glucosamine. Right? It is in two places. Chitin is in two places. First one is it is in fungal cell wall. In where? Fungal cell wall. Funguses also having a cell wall. In fungal cell wall, chitin is there. And chitin is also there in exoskeleton of arthropods. What is the exoskeleton of arthropods? The meaning of exoskeleton. When you see cockroaches, they have a shine exterior. In ants also there's a shine exterior. There's a skeletal which is exoskeletal. We call this as exoskeletal. That's why when a bug dies, what happens? They leave like skeletal, like when a cockroach or some insect dies, you can keep the structure. Other structures will be there for some time. The capsule like thing. පොත්ත වගේ හිටින්නේ ළමයි capsule එක වගේ කැරපොත්ත මැරුණොත් කුරුමිනියෙක් මැරුණොත්. That's the exoskeleton. That gives the shiny appearance to the insects. That's why people can collect insects. Did you have seen any people who are collecting insects? Not live insects, died insects. They collect them. देखला है? सामान्य एकत्र करना देखला आपने? Some keep butterflies, ne? They pin them and keep them. Like that, some are keeping bugs. Like big bugs, small bugs. Bugs they can keep. That's because of that exoskeleton. Right? So it will remain for some time. Right? These exoskeleton. කැරපොත්ත එහෙම දැකලා තියෙනවානේ අර පොත්ත වගේ අරින්නේ. That's the exoskeleton. Right? So that's the exoskeleton. So exoskeleton of arthropods also made up of chitin. The exoskeleton of arthropods. So these are the examples for polysaccharides. So that's what we have to know about polysaccharides. Right. So Lamai, let's go through the short note again. Take the short note of polysaccharides. Right. Take the short note of polysaccharides. See. In polysaccharides, wait a minute. In polysaccharides, what is in polysaccharides? Polysaccharides, characteristics. What are the characteristics? All are macromolecules. Macromolecules means They are polymers and their molecular weight is more than 10 to the power 4 to 10 to the power 5. They all are biopolymers. Few hundred to few thousand monosaccharide subunits. They are not sugars. They are not water soluble. They are in non-crystalline form. Then there are two types of polysaccharides based on their function, and we can categorize polysaccharides according to their function and according to their architecture. According to the function, there are two types of polysaccharides: storage polysaccharides and structural polysaccharides. Storage polysaccharides are starch and glycogen. Structural polysaccharides are cellulose, hemicellulose, and pectin. Based on the architecture, there are linear polysaccharides and branch polysaccharides. What are linear polysaccharides? Cellulose, amylose are linear polysaccharides, and glycogen, amylopectin, and hemicellulose are branch polysaccharides. Then we discuss about the structures. What are they? Examples. What are they? Starch made up of glucose stored in plants. Glycogen made up of glucose stored in animals and fungi. Cellulose made up of glucose component on cell wall. Inulin is fructose made up of fructose. It's stored in tubers of dahlia. Pectin is made up of galacturonic acid in the middle lamella. Hemicellulose made of pentose and hexoses in plant cell wall. Chitin is a glucose. It's made up of glucose amine. It's a nitrogen containing polysaccharide in fungal cell wall and exoskeletal of arthropods. Then we have to discuss about the functions of carbohydrate. Then this carbohydrate part will be over, right? So I'll give you a break, my small break. Now it's 5:20. Let's start the class in 20 minutes. In 5:40, we'll start the class again. Okay. So the online students keep be in the Zoom meeting. Don't leave the meeting. Okay, Lama. Let's start the rest of the class. So we discussed about polysaccharides today, disaccharides and polysaccharides. Then now we are discussing about functions of carbohydrates. Then the carbohydrate part will be over, right? Functions of carbohydrates. Functions. Carbohydrates. Functions. So there are functions of monosaccharides. First, we have to know the functions of monosaccharides. Monosaccharides. What are the functions of monosaccharides? They are energy source. They act as energy source. What's the meaning of energy source? That used to generate energy. What is the most common energy source? Is glucose. Glucose is the commonest energy source in our body, right? Then monosaccharides are the building blocks of disaccharides and polysaccharides. Disaccharides and polysaccharides are formed from monosaccharides. So they are the monosaccharides are the building blocks. Building blocks of disaccharides and polysaccharides. All the disaccharides and polysaccharides are made from monosaccharides. Then monosaccharides are used in forming nucleotides. Do you remember the nucleotide? You learned in O level. Nucleotides are the monomers of nucleic acids. Nucleotides are, they have a pentose sugar. Can you remember? Nitrogenous base and a phosphate group. These are nucleotides. You learn these things in O level, right? This pentose sugar. It's a pentose sugar. Pentose means it contains 5 carbon atoms and it's a sugar. This is a monosaccharide. So monosaccharides are used in for what? Used for producing nucleotides, right? So it's monosaccharides are it is ribose or deoxyribose. We'll discuss about it later. Anyway, you have to remember that it's a pentose. It's a pentose. Pentose means it's a monosaccharide. So monosaccharides are component of nucleotides. Monosaccharides are component of nucleotides. Monosaccharides are components of nucleotides, right? Then disaccharides. What are the functions of disaccharides? And disaccharides. What are the functions of disaccharides? Disaccharides, right? Disaccharides. Lactose. It's a disaccharide. Ne? Lactose. Lactose is the storage sugar in milk. Milk store sugar as lactose. So lactose is the storage sugar in milk. Right? Lactose is storage sugar in milk. Then sucrose. Sucrose is storage sugar in sugarcane. Usually plants store sugar as what? Store carbohydrate as starch. Usually plants store carbohydrate as starch, right? But in sugarcane, carbohydrates are stored as what? Sucrose. That's why we eat sugarcane. If you eat some random plant, does it sweet? No. But sugarcane is sweet. Every part of the plant is sweet. Why? Because they store sucrose as the sugar. So sucrose is storage sugar in sugarcane. Right? Storage sugar in sugarcane. Then it is the carbohydrate which transported within phloem, right? It is translocation in phloem. It's translocated in phloem. Translocation in phloem. So what is the example? So you are making jaggery from what? You are making kitul jaggery from what? Phloem of the kitul tree. You cut the flower of kitul tree. Then what happens is phloem tissue will be cut. Then what is oozing from it? Phloem. Sucrose is the thing. Sucrose is oozing from. Phloem, inside phloems, the carbohydrate is transported as sucrose. That's why we call sucrose as the transporting carbohydrate in plants. We call it as transporting carbohydrate in plants. Storage carbohydrate is starch. Transporting carbohydrate is sucrose. So translocation in phloem, sucrose. Then there's another disaccharide. What is it? Maltose. Actually, maltose is in where? Germinating cells. What are germinating cells? Germinating cells means they are growing. Growing, germinating seeds means, germinating seeds means, germinating seeds means. So this is not, should be not cells, it's seeds. Okay, germinating seeds. What are germinating seeds? Seeds which are producing plants. When you did you eat, did you have ever eat a germinating seed? What? One other kind germinating seeds. Beans. What we do? We we put it in water for 24 hours. Then what happened? It will germinate. It will give rise to a plant. Small plants are starting to grow from the seed. Why we put those seeds in water and then cook it? That's because when the seed is germinating, normally in seeds there are starch. But when germinating, the starch is converted to maltose. Then it gives a sweet taste. That's why we eat germinating beans. Right? Then then have you ever eaten that germinating coconut? Palapol. Palapol kalne? Palapol? Kalne? දැකලා තියෙනවද පැලපොල්? අර some people are beside the road keeping a bag full of balls like whitish balls. මෙහෙම තියාගෙන පාරායිනේ තියාගෙන විකුණනවා. බෝල විකුණනවා. අම්මලගෙන් තාත්තලගෙන් අහලා බලන්නකෝ. Ask from your mothers and mothers, they will know. They are, when the coconut is germinating, when then coconut will give rise to some leaves නේ. මෙහෙම leaves එනවනේ coconut එකෙන්. Then we cut it out. Normally there's a hollow in the coconut which filled with fluid. In here, it's not a hollow. It's like this. It's a round shape, whitish stuff. This is called as germinating coconut, palapol. It's sweet. It's have a different sweet taste. It's different from sugar. It has a sweet taste, but it's different. That's the maltose taste. Right? So in germinating cells, there are maltose. අම්මලගෙන් ඉල්ලගෙන කන්නයි. පාරේ අයිනේ තියෙනවා විකුණන්න දැකලා ඇති. සුදු පාට බෝල වගේ ටෙනිස් බෝල වගේ දාගෙන පාරායිනේ තියාගෙන පාරායිනේ විකුණන්නේ. මෙහෙම අල්ලල්ල පෙන්නගෙන ඉන්නවා. දැකලා එරන් ඇති. එච්චර ක්ලික් වෙන්නේ නැහැ ඕගොල්ලෝ. පළපොල්. හරි? පොඩි බෝල ටෙනිස් බෝල වගේ ගොඩක් දාගෙන ඉන්නේ. Right? So monosaccharides are energy sources, building blocks of disaccharides and polysaccharide, component of nucleotides. Lactose, storage sugar in milk. Sucrose, storage sugar in sugarcane and translocation in phloem. Maltose is germinating seeds. Then polysaccharides. What are the functions of polysaccharides? What are the functions of polysaccharides? Polysaccharides. Polysaccharides. What are the functions of polysaccharides? What are the functions? We already know. What are the functions? Two main functions. Storage, polysaccharides, and structural. What are the storage polysaccharides? Starch, glycogen, and inulin. What? Starch, glycogen, and inulin. starch in plants, glycogen in animals and fungi, glycogen in animal and fungi, inulin in dahlia tubers. Right? Then what are the structural polysaccharides? Cellulose in cell wall of plants and chlorophytes. Then pectin, pectin in middle lamella. Then hemicellulose, hemicellulose also in cell wall of plant cells. Cell wall. Then what else? What else? Chitin. Chitin in exoskeletal of arthropods and fungal cell wall. And there's another one called peptidoglycan, which is in cell wall of prokaryotes. Peptidoglycan, cell wall of prokaryotes. What is prokaryotes? Not having a, yes, not having a what? Developed structure. Well developed nucleus. Nucleus, not structure. Prokaryotes and eukaryotes. Those are the two types. Ne? There are two types of cell organization, prokaryotic and eukaryotic. We will learn it soon, not so long. It's here in the unit, this unit. We learn about prokaryotes and eukaryotes. Okay. Prokaryotes doesn't have a organized nucleus. Examples are bacteria and archaea. All the bacteria, all the archaea, all the cyanobacteria, all are prokaryotes. Eukaryotes are protists, fungus, and plants and animals. Right. These are the functions that you have to keep in mind about polysaccharides. Okay. Right. Then we have to, right, that's all for the carbohydrates. That's all about carbohydrates. Carbohydrates. So carbohydrates, what we discussed about carbohydrates? We discussed about the basic characteristics of carbohydrates. Then we discussed about monosaccharides, disaccharides, polysaccharides, and functions of carbohydrates. That's what you have to learn. That's what you have to remember, right? Right. Now let's talk about the next organic molecule, right? There are main 4 organic molecules in our body, right? There are 4 main organic molecules. What are they? Carbohydrates, lipids, proteins, and nucleic acids. Carbohydrate, lipids, proteins, and nucleic acids, right? So we discussed about carbohydrate. That's all you have to know about carbohydrates, right? That's all you have to know. There are a few more things which we discuss in practical parts, right? There are separate points you have to remember about practicals. Practicals are the same as those practicals. Benedict test and other tests are there, right? So we'll discuss it later. For now, these are the things that you have to remember about carbohydrates. Right? Okay. Now let's talk about lipids. What are lipids? Lipids. Lipids. When talking about lipids, lipids are hydrophobic. Lipids are hydrophobic. What is the meaning of hydrophobic? Phobic means, phobia means afraid of. Afraid of. And there are some diseases called phobias. Some are phobic to snakes. They are everybody phobic to snake. That's not the phobia. Means you can't even see a photo of a snake. There are some people who are phobic to butterflies. When a butterfly comes, they will faint. Right? Then there are a condition called claustrophobia, which means you are afraid of sealed places. If you are put in a small room or something, you will be phobic and you will get anxious and something. Right? They are diseases. Then phobia means you are afraid of. There's a condition called social phobia. Social phobia means you are afraid to, it's a pathology. It's not normal, okay? You are phobic to face people. So you always hide in the house, you don't go out, you are afraid of people, human. Like that, their phobia means afraid. Hydrophobic means, hydro means water. They are afraid of water. That's why it's called as hydrophobic. When you put coconut oil, it's fat. It's a lipid. Coconut oil to water, what happens? Does it mix? It doesn't mix. They don't mix. What happens? They will be separated from two layers. The reason for that is they are hydrophobic. They don't dissolve in water. To dissolve in water, what should happen? Huda. To dissolve in water, what should happen? What should be there? What molecules dissolve in water? What are the molecules which dissolve in water? What is the characteristic the molecule should have to? They should be polarized. They should be polar molecules. If not polar, they should be ionic molecules. So these lipids are insoluble in water, right? They are insoluble in water. They don't dissolve in water. Sometimes, but sometimes they dissolve in organic solvents. There are some organic solvents which are non-polar. You have to remember this. Polar things will be dissolved in polar solutes will be dissolved in polar solvents. Non-polar solutes will be dissolved in non-polar solvents, right? So the water is polarized, but lipids are not polarized. So they don't dissolve in water. But lipids might be dissolved in some organic solvents, which are non-polar, right? Right. Then they are large biological molecules. They are large biological molecules. Even though they are large, they are not considered as macromolecules. Even though they are large, actually they are large. Their molecular weight is more than 10 to the power 4 to 5. But we don't consider them as macromolecules. Why? Because they are not polymers. They are not polymers. They are not polymers, right? They are not polymers. To become a polymer, what should happen? Monomers should attach together like this, ne? But here they don't polymerize, right? So lipids are not polymers. So lipids are not macromolecules, not macromolecules. They are not macromolecules. They are not polymers. They are not macromolecules, right? You got it? Right. Then elemental composition is of lipids is same as carbohydrate, OCH. It just contains oxygens, carbons, and hydrogens only, right? There are no nitrogens, no sulfur, and no other phosphorus or other things, right? Hydrogen to oxygen ratio in lipids are not 2 to 1. It's always more than 2 to 1. That means why hydrogen and oxygen are 2 to 1? Because in carbohydrates, always when there's one carb oxygen, there are two hydrogens in carbohydrates. But here it's not like that. Always there are more hydrogen than oxygens. Always there are more hydrogen than oxygen. The reason is like this, because in, can you remember, there are two components in lipids. What? Glycerol and a fatty acid. Glycerol and fatty acids. So fatty acids, what is the structure of fatty acids? It's like this. It has a carboxyl group, C double OH in a end, one end. Then they have a long carbon skeleton, right? Long carbon skeleton, like this. In this carbon skeleton, there are hydrogens attached to it. Hydrogens attached to it. What happens? There are so many hydrogen molecules. In here, there's only 1, 2 oxygens. But how many hydrogens? Plenty of hydrogens. Then there won't be the ratio of 2 to 1 in lipids. Right? There won't be a ratio of 2 to 1 in lipids. Always more hydrogen than oxygen. Right? There are 3 biologically important types of lipids. There are 3 biologically important types of lipids that you have to know. They are fats and oils, and phospholipids and steroids. Those are the important types of proteins, biologically important types of proteins, right? Those are the biological important types of proteins, right? So I'll do a quick recap of what have we done so far, right? Just follow me. I'll give a separate recording of it, right? Quick recap. So you just go through, even though you are in the class, just go through it. It will tell everything that we have learned in unit 2, right? Usually I do it in other classes, like revision classes, I give a recap. So in the recap, I explain everything that I've done on that day in the class. But everything we have done on today is very small part. So it's the beginning. So we are doing very, very small parts only. So there's no point of giving a recap of what have we done today. I'll give a recap of what have we done in the unit 2, right? I'll tell everything. Just follow the short note. Take the short note and follow it. Okay. Right. So I'll give the recording of this video. So you just go through it, right? It will be 10 minutes, like 10 minutes. When if you go through it, you can memorize all the things what we have done in unit 2 up to now. Okay. Right. So